So our next speaker is Coy Allen. He's an assistant professor in the Department of Biomedical Sciences and Pathobiology here at Virginia Tech as part of the Virginia-Maryland Regional College of Veterinary Medicine. He began there in November of 2012. He got his BS in biology from East Carolina University, MS in biology from the University of North Carolina at Greensboro, and PhD in genetics and molecular biology from the University of North Carolina at Chapel Hill. And then, interestingly, in addition, and perhaps he's thinking ahead in terms of science management and entrepreneurship and so on, but in any case, he got his MBA with a concentration in biosciences management from North Carolina State University. He did postdoctoral work and then was a research assistant and research associate professor at the Lineberg Comprehensive Cancer Center at the University of North Carolina in Chapel Hill. His research interests are in innate and adaptive immunity and host defense against pathogens and cancer immunology. And his talk today is epithelial cells and their role in immunity.
All right, thank you for the introduction. Can everybody hear me? Yeah? All right, get grins and nods in the back. It's always a good sign. Hope everybody's enjoyed the presentation so far. I hope to continue the trend. Today I'm going to be talking a little bit outside my comfort zone. This is something that we work on quite a bit in my lab, epithelial cells. I'm also going to be a little bit more basic than some of the talks you've probably heard today. That's just to bring you all on board to a cell type that you probably aren't used to working with. Now, I think we all kind of have a concept of what epithelial cells are. However, what you typically don't think of is a role for them in the innate immune response. They are critical. Here at the top is are epithelial cells that we grew up in our lab. If you can see, they're actually very pretty cells, right? They're very complex cells. When we grow them up in the dish, we can take them from mucosal sites, typically the gut or the lungs. We can grow them on a dish, and over the course of a few months, well, not a few weeks, it usually takes about 6 weeks, these cells will begin to polarize. They'll begin to form their cilia. Cilia will begin to beat, and eventually the cilia will begin to beat all all together, which is actually a kind of neat process, and it's a very powerful tool when you're trying to validate models associated with mucosal immunology. In this case, the green at the top here are lenticular cilia, and the nuclei polarize down to the bottom of the cell, called the basolateral surface. So today we're going to be talking about a few aspects of the epithelial cells that can affect mucosal modeling. Okay? The first thing we're going to discuss are some of the secreted factors. Epithelial cells produce mucus. They come in two different types that we're going to discuss today. We have the mucus-producing cells, and we also have the ciliated cells here as well. The ciliated cells are the ones you're probably most familiar with. However, they can differentiate into goblet cells, which are the ones responsible for mucus production. In addition to mucus, these cells secrete type 1 interferons, and they also produce very potent antimicrobial peptides known as beta-defensins, for example, are a good example of those peptides. They can also produce reactive oxygen species, reactive nitrogen species, so they're very potent. They produce very potent mediators that can affect the microbes that are trying to invade, pathogenic or illness in the microbiome. They also provide, as we know, a physical barrier that protects the mucosal tissues. They can also be active participants in the immune response by secreting cytokines and chemokines, and that's what we're going to spend most of our time talking about today here, is to recruit other white blood cells to the site of infection. They are very pretty cells to look at. Again, this is they typically make the cover of journals when you work with them. You always should take a scanning electron micrograph picture because you never know when you might need it. Now, the first thing I'm going to talk about are some of the secreted effectors. As I discussed the mucins, if you look at the mucins, look at that. It's just a gemisch, right? It just looks sticky. Mucins and mucus production are what we typically think of when we have an infection, right? We blow our nose, right? We have our mucus production. Looking at this, though, you can see that this is a very effective barrier. Bacteria, pathogens, elements of the microbiome have a hard time migrating through this web of mucus. Mucins are produced by MIEP genes. These are driven by NF-kappa B signaling. We'll talk about a little bit about NF-kappa B signaling as we move forward. NF-kappa B is a master regulator of gene transcription. We usually think about it for its role in driving cytokine and chemokine production. But you also be aware that it influences a lot of aspects of the mucosal of the epithelial cell biology, including mucus production. The antimicrobial peptides are another very potent defense mechanisms that epithelial cells utilize. There's a lot of work right now studying antimicrobial peptides because they're so effective at basically killing bacteria. Because bacteria are developing antibiotic resistance at an increasing rate, a lot of companies have sprung up developing antimicrobial peptides, particularly beta defenses, as an alternative approach to classical antibiotics. Epithelial cells also produce immunoglobulins. Right? They secrete or release quite a bit of immunoglobulins into the mucus, or at least they allow the passage of these immunoglobulins. Typically, you're talking about IgE into the mucus, and this, of course, is free tagging all the epitopes and all of the antigens in the mucus, so that it makes the presentation much more effective. We're not really going to discuss the immunoglobulins. We are going to talk quite a bit more about mucus. So some of my work involves pattern recognition receptors, which we're going to get to. And one of the effects of activating pattern recognition receptors is the activation of beta-catenin B. Beta-catenin B is activated. In addition to producing cytokines and chemokines, we also get mucus production. These are epithelial cells. This is the epithelial cells layer from the intestine. These are samples I did last week, as a matter of fact, so just in time for this talk, right? So we prepped some of these with Alcyan blue periodic acid shift staining. And it produces a very pretty picture of the goblet cells and mucus in the gut, and also the lungs. This is classically used in the lungs, but we modified it to function in the gut as well. What you're looking at are the dark blue circles here. These are the dark blue areas are the goblet cells. The light blue areas are the mucus. Now, we flush our colons out when we prep them, so if we didn't flush them out, you would have a much denser light blue area around the tops of the crypts here, around the tops of the filament. Now, when we challenge these mice with pathogens, with DSS, which is shown here, that's dichloride sulfate sodium, this is inducing colitis, we cause mucus hypersecretion, cause inflammation. And as part of this, you can see that the animals that were challenged with AOM, which is a tumor-inducing agent, and DSS, which have inflammatory bowel disease, basically, they produce more mucus. They have more goblet cell hyperplasia compared to our mock-treated mice, which are under control condition. So mucus is really important. Mucus helps prevent bacteria and pathogens from reaching the epithelial cell layer. Now, a lot of the animals that I work with that are lacking pattern recognition receptors also lack the ability to produce mucus in significant numbers, or in significant volume. And the problem with this is that it allows bacteria to get into areas of the crypts that have become inaccessible, and it creates a niche for the bacteria to overgrow. So a lot of the pattern recognition receptor mice that I work with that lack pattern recognition receptors actually have problems with specific bacteria that overgrow because they are allowed to gain access into regions of the colon that they wouldn't normally be able to gain access to. Now, this is just a diagram just a schematic of how mucus works, right? We don't usually think about all the dynamics of mucus, but there's actually an entire field of science that studies this, okay? The mucus layer in the gut and in the lungs are typically somewhere between 10 and 100 microns in depth, and this allows just a really nice overview of how mucus works. It keeps most of the bacteria and elements of the microbiome at the top. The secreted factors tend to be more in the middle to bottom areas of the mucus, close to the epithelial cells. Now, we have problems when we encounter pathogens that are able to navigate this mucus. A lot of pathogens can actively swim through the mucus, basically, because they're flagellated. Salmonella is a good example. There's some other bacteria that are able to produce enzymes and proteolytic enzymes that are able to break down the mucus, that allow the bacteria to literally just digest its way down to the epithelial cells. Once at the epithelial cells, then that's where the epithelial cell itself will encounter the pathogen. At that point, the epithelial cell now becomes an active participant in the innate immune response. Okay? Now, by active, I mean these cells express classes of pattern recognition receptors. Now, my lab studies these three classes of pattern recognition receptors. These are I would argue these are the main classes. There are some others. For example, C-type lectin receptors aren't up here. But these are, I would say, the best characterized. These happen to be the ones I worked on as well. So our lab studies toll-like receptors, reggot-like helicase receptors, and NOD-like receptors. We study these in the context of epithelial cells and also in the context of the immune cells themselves, mostly macrophages. And so a lot of what I'm going to tell you today that's associated with epithelial cells also apply to macrophages and other leukocytes as well. Now, the toll-like receptors here are probably the best characterized of the pattern recognition receptors. I think everyone in here probably is familiar with the toll-like receptors, or at least you've heard of them. And for the purposes of today's talk, I've simplified these signaling pathways down to sort of their essence. Okay? So following toll-like receptor activation, the majority of toll-like receptors signal through the MIE-88 adaptive protein. And the net result of toll-like receptor activation is the activation of cytokine genes due to NF-kappa B activation. Again, this is the master regulator to transcription. Now, I'm going to tell you about these other two pathways, but I want you to understand that all three of these pathways interact with one another. It becomes a very complex web. However, after seeing some of the talks today and through this conference, I can see that interacting webs aren't a problem for you guys, right? So that's the whole point of computer modeling. You're generating all those webs of interaction. But for today, I wanted to simplify it down to its essence. Okay? Much more complex. Now, the toll-like receptors activate NF-kappa B. What about these other two classes of pattern recognition receptors? The next class we're going to discuss is the ring-like helicase receptors. These are almost, I would be surprised if you've heard of them. However, they are a very interesting family of pattern recognition receptors that are responsible for sensing viruses. There are two main members of these the ring-like family, the ring-I and MIEP. These are responsible for sensing very specific sets of viruses. Right? They sense basically viral RNA or viral nucleic acids. Then they interact, once they sense these pathogens, they interact with MABs. MABs is the mitochondrial antiviral signaling protein. As the name would suggest, MABs uses the mitochondria as a scaffold to drive the innate immune response. It's a very interesting field that we're working into. Over the last 5 years, there's been a lot of work characterizing these receptors, how they interact, and how they use the mitochondria as a scaffold. This is really important in the context of epithelial cell biology, because if you remember, I told you that the epithelial cell is polarized. Well, one part or one aspect of that polarization is the migration of the mitochondria in the cells up to the cilia. If you think about it, the nucleus translocates down to the basolateral surface, the mitochondria translocate up to the apical surface, which is right under the cilia. Well, there's two reasons for this. The first reason is that the cilia need energy. So the mitochondria are right there to provide energy to the cilia to beat. But if you think about it, by being tethered to the mitochondria, by using that as a scaffold, we've evolved this mechanism to place the pattern recognition receptors directly under the point of entry for viruses when they come into epithelial cells. And this is the case for viruses that we study in our lab, RSV and influenza, which come into the cell right at the epithelial cell apical surface, right below the cilia, and they're immediately sensed by these scaffold, these proteins that are used in mitochondria as a scaffold. Following activation, the ring-like helicase receptors drive IRF transcription, drive the transcription of type 1 interferon genes through IR3 and IRF7. And then the last group of pattern recognition receptors we're going to discuss are the NOD-like receptors, NLRs. Right? NOD-like receptors are just an easy way to pronounce the name. The real name of these are the nucleotide bonding polymerization domain containing leucine-rich repeats receptor protein network. Right? So huge name, right? So we're just going to call them NOD-like receptors, or NLRs for the rest of the talk. There are several functional groups of NLRs. There are 22 NLRs that have been characterized in humans and other mammalian species. Of those, only about half are well characterized. They've been identified but aren't very well characterized functionally. And of these, the best characterized subgroup are the NLRs that form a multi-protein complex, which is known as the inflammasome. This is a large multimeric protein complex that comes together upon activation of the NLR or the sensing of a pathogen. And they form this inflammasome, which is responsible for the processing and cleavage of pro-IL-1 beta and pro-IL-18 into their mature cytokines. So these two cytokines are really potent regulators of the immune response. They drive a lot of aspects that we think of when we think of being sick. For example, IL-1 beta is a pyrogenic enzyme, cytokine. So when you have a fever, it's because you have high levels of IL-1 beta, among other cytokines. Right? So these are really potent mediators of the innate immune response. IL-18 is really important in the context of the mucosa because IL-18 is a very potent cytokine. It's also a very potent moderator or modulator of epithelial cell regeneration and repair. We'll talk about that a little bit more as we move forward. All right, so the epithelial cells, when they engage pathogens, when they engage elements of the microbiome, it's really important that they maintain a balanced response. They're always coming in contact with elements of your lumen, okay? So it's really important that they not immediately signal an immune response. And they do this through a couple different ways. One is physically locating the physical location of the pattern recognition receptors. In this case, we're talking about the toll-like receptors. In general, toll-like receptors, we can consider those as being extracellular versus the NLRs, the read-out-like helicases, which are cytosolic. Okay? There are some toll-like receptors that are intracellular, but for the purposes of today, we're just going to focus on the extracellular receptors. And if you look, this is a really nice diagram that shows the epithelial cells here. It shows the base of the crypts here with the Pannin cells. This is the basolateral surface of the epithelial cells on the bottom. The apical surface are the ciliated areas at the top. Okay? And the lumen is here, and then the tissue itself is here. Okay? Now, if you look at these cells, they orient their pattern recognition receptors on the basolateral surface in general. Most of the epithelial cells that express high levels of TLRs, they're oriented on the basolateral surface. This is a physical way that you can restrict the initiation of the immune response. Because bacteria connecting here at the cilia won't activate the immune response, but if you have a barrier breach, if you have a chronic condition, for example, if you have epithelial cell damage, and you have bacteria that translocate from the lumen into the tissue, that places these pattern recognition receptors now at a very good point where they can interact with these pathogens, interact with these microbes, and drive the immune response because they know that the pathogens or the microbes have now entered an area of the tissue where they shouldn't be. So that's a very nice physical way that the cells can orient these pattern recognition receptors that can maintain some level of balance. Now that's not the case with all of the epithelial cells. There are some that differentiate, particularly near the Peyer's patches, that do have some toll-like receptors. TLR2 and TLR5 are probably the best characterized. These are usually near the M cells. M cells are specialized phagocytic cells that allow antigens to pass from the lumen to the Peyer's patch area, so it allows antigen presentation. And because of this, this is sort of a weak point in your defense system. And so it's thought that the epithelial cells that surround that need to really bulk up their defenses. And so by doing that, they express some of these pattern recognition receptors on both sides. This allows for the host to maintain some low level, some baseline level of inflammation. It allows recruitment of leukocytes to the Peyer's patches. So you have this baseline level, and that's one way that the epithelial cell balances. But I will tell you that the expression of toll-like receptors on that apical side is rare. It only happens in very specific conditions. Now, once you have a barrier breach, so here we're showing the barrier breach, so we've had some sort of damage to the epithelial cells. We have bacteria that translocate from the lumen into the mucosa tissue. Those bacteria are recognized by a diverse repertoire of toll-like receptors on both the leukocytes as well as the epithelial cells. And we've done quite a bit of studies looking at toll-like receptor knockout animals, looking at MIE88 deficient mice. So when you knock out MIE88, you're basically knocking out almost all of the toll-like receptors. So you knock out everybody except TLR3. We've also done several studies looking at TLR2 and TLR4 specific knockout animals and different models of infection. So looking at various viruses and bacteria, as well as in the context of inflammatory bowel disease, and also in the context of asthma as well. And in all of these situations, the moral of the story is basically the same. If you don't have toll-like receptor signaling, you tend to have a reduced production of NF-kappa B activity. Right? So in general, I'm being very general here. But when you lack NF-kappa B, you lack the pro-inflammatory cytokine signals. And in the context of bacteria, what that allows, it allows the bacteria to make inroads into the tissue. So the bacteria are able to migrate much farther beyond the epithelial cells into the tissue, eventually entering the bloodstream. And what we typically find when we're looking at bacteria infections in these knockout animals is that we tend to get a systemic infection much sooner. In general, as you would predict, when you knock out your elements of your innate immune response, it becomes very hard to control bacteria and viruses. And what we typically end up with are are cases of sepsis, where bacteria make it into distal organs such as the liver, the spleen, and ultimately the animal has to be euthanized. So in the context of inflammatory bowel disease, or in the context of knocking out these genes in the gut and then inducing inflammation, usually using the DSS, we also see that this is detrimental to both the epithelial cells as well as the leukocytes. We've been able to show using different models, for example, bone marrow chimera studies, and different or cell type specific knockouts in these pathways, that the epithelial cells play a vital role here as well as the leukocytes. And in general, when you knock out these receptors, you tend to get more chronic inflammation as well. So it has to do with the bacteria being able to constantly signal through other pattern recognition receptors and making it past this barrier into areas where it should be, sort of maintaining this low level of inflammation. Now, the inflammasomes are a little bit different story. Okay, this is really one of the main focus areas of my lab. We study NLRs in the context of inflammasome-forming NLRs, as well as other functional groups of NLRs, which we'll discuss in a little bit more detail. This pathway may look a little confusing, again, but I'll try to summarize it. So what we have are inflammasome-forming NLRs here. These are cytosolic receptors, and so we're sensing intracellular pathogens, intracellular stress, and damage, which can cause intracellular changes. Different NLRs sense very specific stimuli. For example, there's an NLR that senses flagellum, so it responds to flagellated bacteria. There's another NLR, NLRP3, which is shown here, which is the best characterized NLR because it's the most promiscuous. It seems to be activated by a whole suite of stimuli. Following activation, the NLR will undergo conformational change. In this case, NLRP3 can be activated by, we've looked at, my own lab has looked at a wide variety of DAMPs. We've looked at potassium and calcium. We've looked at reactive oxygen species production, LPS, poly-IC. All of these seem to activate NLRP3. The way that it seems to be activated is associated with changes in the intracellular environment rather than sensing a specific ligand. And so following activation, it undergoes a conformational change, where this long string of leucine-rich repeats is normally folded over the nucleotide binding domain and the pyrin domain, which is here. So we usually have this folded protein, but upon activation, it opens up. When it opens up, it allows binding and oligomerization to other NLRP3 molecules, which have also been activated, forming this large oligomeric structure. At the same time, we have the formation of what's known as the ASC spec. ASC is an adaptive protein that facilitates inflammasome function and inflammasome activation. And this spec and this NLR function together to recruit and activate caspase-1. The net result of NLR inflammasome activation is the activation of caspase-1. Caspase-1 is a caspase. It cleaves molecules. In this case, caspase-1 specifically targets pro-IL-1 beta and pro-IL-18 for cleavage, and you then have the maturation of these cytokines which get cleaved. Now, at the same time, caspase-1 is also associated with a very novel form of cell death known as pyroptosis. Again, when you're talking about the epithelial cell layer, that's really important because you want to avoid epithelial cell death at all costs. And pyroptosis is a very specialized form of cell death. It's a very controlled form of cell death, mediated by caspase-1. We'll talk a little bit about that also as we go forward. Now, in addition to what's known as the canonical inflammasome activation shown here, we also have activation which is driven by caspase-11. This is a very new hot field in the inflammasome world. We are working on caspase-11 now. We just recently submitted a manuscript for caspase-11. So this activates what's known as the non-canonical inflammasome. This is very interesting work. Turns out that a lot of the previous studies looking at caspase-1 were using animals that were actually caspase-1 and caspase-11 double deficient. So we just found that out back in 2011. So a lot of studies have now gone back to try to figure out, tease out the differences between caspase-1 biology and caspase-11 biology. And the non-canonical inflammasome is a major mediator of pyroptosis, but it can also feed back. It's been shown to be elements of non-canonical inflammasome signaling have been shown to be essential for driving the canonical inflammasome. There's a lot of work trying to tease these two mediators out. So it's a very complex system. we have a very limited repertoire of NLRs that sends a whole suite of pathogens and elements of the microbiome. These NLRs signal through a diverse range of signaling pathways, ultimately leading to the formation of the inflammasome. And a lot of this isn't well understood. And that's one place where I think computational modeling can really help in this field, to try to tease out what drives this non-canonical inflammasome versus the canonical. What are the ligands that these NLRs are recognizing that their computational changes? Now, there are only 6 of the 22 NLRs that have been characterized as forming an inflammasome. Somehow these 6 are responsible for recognizing the whole repertoire of bacteria and elements in microbes. So it's a very complex system that we don't really fully understand. So, in the context of intestinal homeostasis, the NLRs have been shown to be vital. Some of my own work is summarized here. We've shown that elements of the NLR inflammasome are essential for maintaining intestinal homeostasis. And when we started these projects, we assumed that when we knocked out elements of the inflammasome, caspase-1, PICARD, or ASC, and different NLRs, we would have reduced inflammation because we would have reduced IL-1 beta and IL-18 levels. And sure enough, that's what happens. When we knock out these genes and we challenge the mice in models of inflammatory bowel disease, we get reduced levels of these 2 cytokines. Yet we get increased disease. We get significantly increased colitis. These mice have significantly increased tumorigenesis. So in the inflammation-driven tumorigenesis model, the mice are worse off. So how could that be? We've knocked out a major pro-inflammatory mediator. We've knocked out 2 very potent pro-inflammatory cytokines. Well, it turns out that the secret lies with IL-18. It took us a couple of years to figure this out. So we were originally approaching this that IL-1 beta and IL-18 were pro-inflammatory cytokines. But it turns out that IL-18, by being a potent modulator of epithelial cell regeneration and repair, turns out that loss of that trumps loss of the pro-inflammatory cytokine effects. So what happens when we are working with mice lacking elements of the NLR inflammasome, likewise mice lacking MIEP, which is important for gene transcription to generate the proforms of those cytokines. Turns out when we're working with mice or animals lacking these genes, we're getting reduced IL-18, and we're also getting reduced epithelial cell regeneration and repair. So this is causing damage to these epithelial cells. This is allowing more bacteria to translocate, reaching deeper areas of the mucosal tissue, signaling through other modulators such as IL-6, TNF-alpha. Those levels tend to increase in these animals. So by lacking this epithelial regeneration repair process, these epithelial cells are much worse off, allowing more bacteria to translocate, maintaining this very low level of inflammation all the time in the gut. Right? This causes a lot of neutrophils to be recruited, and over time those neutrophils cause more and more damage. In the context of tumorigenesis, what we've actually found is that the deficient repair processes will eventually, over the course of a few months, result in extensive colitis-associated cancer. So you have increased inflammation, you have increased bacteria translocation, you have reduced mucus. Remember I was talking about mucus earlier? You have reduced mucus, so you have bacteria able to enter areas that they normally wouldn't be in the gut. And you maintain this persistent infection, you maintain this persistent inflammation, which drives the disease pathophysiology. NLRP6 is probably one of the best characterized NLRs associated with the epithelial cell process here. The NLR that I work on, the NLRP3, is predominantly associated with macrophages. We've been able to look at, using bone marrow chimeras, we've been able to look at these different genes in tissue-specific contexts. And NLRP6 is expressed in epithelial cells and controls that aspect. Under normal conditions, we have a healthy gut microbiota and engages the NLRP6 inflammasome, which drives mature IL-18 production, leading to interferon gamma. It also drives this regeneration repair process. However, in the absence of NLRP6, you don't have the NLRP6 inflammasome. What happens is that these epithelial cells begin to release CCL5. CCL5 is a very potent neutrophil recruitment cytokine, or chemokine rather. And when you have potent levels of CCL5 and neutrophils coming in, again, the neutrophils cause extensive collateral damage to these mice, and that will drive the inflammation. On the flip side, when we lack NLRP3 or the NLRP3 inflammasome in the macrophages responding to these sites of inflammation lack the ability to produce IL-18 And again, you have that deficient regeneration repair process. Very similar net results in both cases. One is driven by epithelial cell component part, and one is driven by the macrophage. Also, when you remove ASC or caspase-1, either one of these mice are highly susceptible. They're hypersensitive to inflammation-driven tumorigenesis and any gut infection, any gut pathogen, because they lack the ability of both the epithelial cells and the macrophages to drive these processes. So they're significantly more hypersusceptible. Now, the other aspect that I'm just going to hit on here is pyroptosis. This is a very distinct form of cell death. It's distinct because it's driven by caspase-1 and/or caspase-11. It's much more controlled than necrosis. This is a much more controlled cell death. What we think occurs here, we think pyroptosis is a major mediator of epithelial cell denuding. When we infect, for example, mice with influenza that lack elements of the inflammasome, we can see reduced levels of pyroptosis. What we also see are increased levels of epithelial cell damage and stress. And so one of the ways that epithelial cells can combat viruses, sort of the last resort, is a denuding process. When the epithelial cell is infected with a virus that it can't clear, one of the processes is to enter this controlled cell death process where it denudes from the underlying tissue. If you look in the airways, a lot of times you'll see areas that lack epithelial cells, especially following influenza. That's an area we look at quite a bit. You can see these epithelial cells have come off in sheets. Likewise, when you get a pathogen in your gut, a lot of the times the diarrhea you have is full of, you can clearly see your epithelial cells. They've shed the underlying layer. This is a last resort for epithelial cells to get rid of virus. So if you think about it, they become this pocket of replicating virus. So what else is left to do except release yourself from the underlying tissue and be excreted through coughing, through diarrhea, through sneezing, whatnot. Just get out of the system. And that's actually a very effective means to get rid of the virus, but then you leave your underlying tissue exposed. And a lot of times when you're looking at histopathology slides for these samples, you can see white blood cells filling in behind these areas of denuding. So you'll see concentrations of leukocytes under these areas of denuding airway or gastrointestinal. And pyroptosis, we think, is a major mediator of this process. Now, like I said, NLRs are very specific. So they sense very specific aspects of different bacteria and viruses. The ones we've looked at NLRP3, NLRC4, NLRP6, well, I guess we've looked at all of them. We published on these, the NLRP3, NLRC4, and P6. We are about to publish here, and I have some background information on this one, but I'm not going to say anything about that today. NLRP3 and NLRC4, again, these are examples of specificity for the NLRs. NLRC4 recognizes flat flagellum, so it recognizes flagellated bacteria. NLRP3, again, recognizes much more robust signals. We think it senses changes in the intracellular composition of different factors. Right? So mostly potassium and calcium. Again, NLRP1, we've also shown has an effect in these processes. So far, it only has been shown to recognize anthrax and anthrax lethal toxin, and also toxin Gandia. But we've shown that we're through the process of publishing this, that it also affects inflammatory bowel disease, as do really all of these. And it's because they sense different elements of the microbiota, different pathogens, that we really, each one senses very specific, forms a very specific inflammasome in response to a specific stimuli. So it's not all that surprising that each one of them, if you remove it from the system, you get a different overgrowth or different translocation of bacteria, but the net result is the same. They're still more sensitive to the disease process. Now, in addition to driving the immune response, pathogens also have evolved to modulate the inflammasome. Right? Pathogens have evolved elements to functionally inhibit these innate immune responses. For example, NS1, the flu NS1 protein, which is here, directly affects caspase-11. Again, I told you caspase-11 is important for pyroptosis. The flu is it is very disadvantageous for the flu for epithelial cells to denude. So it's reasonable to think that the reason it's targeting caspase-11 is to stop that pyroptosis process, so you keep the airway epithelial cells attached a little bit longer. There are lots of examples of the fluid S1 protein, though it's a very nice example of a pathogen-associated modulator of inflammation. It affects RIG-I-like helicase receptors as well. And this one targets RIG-I, which is the primary pathogen recognition receptor responsible for sensing. It targets the RNA processing of that pattern recognition receptor. So there are lots of examples of pathogens that can modulate the function of inflammasomes, not only inflammasomes, but all the other pattern recognition receptors. All right, so those are the pro-inflammatory pattern recognition receptors. The toll-like receptors that are pro-inflammatory, the knot-like receptors that are pro-inflammatory, right? I'm going to switch gears a little bit and talk about some of our work looking at inhibitory NLRs. This is a new field, a new direction. As I said, there are 22 different NLRs. We've only really characterized half of them. The more we characterize them, the more we uncover some very unique properties in the ones that have yet to be fully characterized. In this context, we have the inhibitory NLRs. There are 3 of them so far that we've characterized that fall into this functional subgroup. NLRP12, NLRC3, and NLRS1. Now these inhibit inflammation. They function through diverse mechanisms, but they all seem to converge on NF-kappa B signaling at some point, which is shown here. NLRP12 inhibits the non-canonical NF-kappa B pathway through interacting with NIC, TRAP3. And it also affects the phosphorylation of IRAK1. Likewise, NLRC3 and NLRS1 also seem to interact with and inhibit TRAP6. So it's a very effective pathway. If you want to inhibit inflammation, target NF-kappa B. Yep.
No direct interaction with
So far, we do not see any direct interaction. It's affecting. As a matter of fact, the hypothesis is that these NLRs form what's known as a trapezoome. So I talked about the inflammasome. It's a multi-protein complex. These NLRs are really good at forming multi-protein complexes. So far, all of the inhibitory NLRs have shown an affinity to trap molecules, be it TRAP2, TRAP3, or TRAP6. That's a very effective way at inhibiting a pathway upstream and having a far-reaching effect. Now, why would you want to inhibit inflammation? Well, inflammation is great. When you get infected, when you're exposed to your microbiome, you want a robust inflammatory response. You want to clear that pathogen away. The problem is after the pathogen is gone, you have to resolve your inflammation. And we think these inhibitory NLRs are key bottlenecks or key mediators of this inflammation resolution. They dampen overzealous immune responses. So likewise, when you don't have them, most of our work has shown that when you don't have these inhibitory NLRs, you have hyperinflammation. You have hyperinflammation in the gut, you have hyperinflammation in the lungs. You were able to control pathogens moderately better. The problem is that you can't shut down your immune response. So you end up getting lots of hyperinflammation in the tissues. And again, you have that collateral damage associated with it. Now, they are much more complex than what I've just shown. They all converge on NF-kappa B signaling, but they also go off and do other things. NLRS1 is an excellent example of this. NLRS1 has been shown to negatively regulate NF-kappa B. It's also been shown to negatively regulate the type 1 interferon response. NLRS1 interacts with MAPs, so it inhibits the binding of RIG-I to MAPs. So this is particularly important when you're exposed to viruses. Following activation, RIG-I or NLRS1 senses the virus at some point and will release MAPs to allow RIG-I binding. This will drive the production of type 1 interferon 6. However, in addition to functioning as a negative regulator, NLRS1 has also been characterized as having some positive regulatory effects as well. NLRS1 has been shown to function by interacting with the protein UQCRC2, which is, I had to look it up, but it's important for reactive oxygen species production, okay? So NLRS1 can positively regulate the production of reactive oxygen species. It's also been shown to function with TUFM, which is a part of this autophagy complex, so it's also been shown to be a positive regulator of autophagy, right? Very complex, and what we think, our hypothesis for why it has all these different functions is that it's very cell type specific, it's very temporal specific, it's very pathogen specific. NLRS1 is a very broad-reaching protein, as are the other NLRs in this group, and so it's very important that they are tightly regulated. It's also very important that they are effective at the pathways that they target. So what we're seeing is that they tend to affect these pathways very high up, allowing shutdown of the final production. I know this is the case in the context of pathogen infections. We've also looked at NLRP12 in the context of colitis and colon cancer. And as you would expect, in the absence of NLRP12, which is here, we have increased non-canonical inflammasome signaling. This results in significant downstream effects, and what we've actually been able to show is that in the absence of NLRP12, we lose NF-kappa B signaling or non-canonical NF-kappa B signaling. This causes increased inflammation, increased tumorigenesis, and this is the main effect of this is that we lose CXCL12 and CXCL13 production, which are important for driving the chemokines that are highly associated with cancer. And so when we lose those, we lose those chemokines, which affects the tumorigenesis aspects. So these are very interesting proteins, very interesting family that we're working on, we've been able to make quite a bit of progress. All right, so I realize this is a diverse group of students, right? So diverse group of people. We've got computational people that have looked at me with just dead eyes, and we've got immunologists that have looked at me with interest and respect, maybe. We'll see. All right, but I'm not going to play the video because we're going to be a little short on time, and I don't want to get into it. But this is a very nice video. It's Nature Immunology production. All right. This will cover mucosal immunology from start to finish in a very simplistic terms, but it's great. It's a really good video. Copy down your web address here. All right. In your spare time, take a look at it. It is a very nice production. All right. If you beg me at the end, I might play it, but we'll see. All right. But I highly recommend it if you're a little bit uneasy with the immunology side. Right? You've heard from folks about T cells. You've heard from me about macrophages and these epithelial cell things. Right? So I highly recommend it. All right. So how can computational modeling help us? I hope you got the impression that we don't really know a lot of things about these pattern recognition receptors. They've been around for years. They've been around. The toll-like receptors have been studied for 40 years. Right, in one form or another. But we still don't fully understand mechanisms. The NLRs are a decade old, and we only have, I hear somebody's already started. Fantastic. Woo! All right, so the NLRs have been around for about a decade, and we still haven't characterized all of them. We have found the low-hanging fruit, the ones that affect iron beta IL-18 production. Very low-hanging fruit. You have a very defined pathway. You have a very defined output. So it's no wonder that they're the best characterized NLRs. So what we hope is that computational modeling can begin to link a lot of these complex interactions between immunity, metabolism, and microbiota, and provide us with a way to look at these pattern recognition receptors as therapeutics. Okay? Do we want to activate them? Do we want to inhibit them? In all of our animal studies, we've shown that when we inhibit them, that's a very bad thing. Mice get really sick. But when we activate them, can we enhance their activities? Can we make them more efficient at what they do? So that's one thing that we're interested in looking at. So making links between these complex microbiota elements, metabolism, immunity, all these different functions. We hope that we can use computational modeling to draw this together. We want to be able to design therapeutics, right? We want to be able to look at these pattern recognition receptors using databases and use structure-based virtual screening to look at large libraries of compounds that could activate these receptors or inhibit these receptors. I can certainly see advantages to both. All right, so one of the issues with the pattern recognition receptor family in general are that we have these great human genetic studies, these great genome-wide screening results, showing different subpopulations with all these mutations in these genes. They're all associated with disease, for example, inflammatory bowel disease. The problem is that for every study we have showing an association, we have 2 studies that show they're not associated. So we end up getting conflicting data. We end up getting lots of conflicting results. So we hope that computational modeling can help reconcile some of these conflicting results, both in population studies as a whole, but also in the NLR and TLR fields themselves. A lot of these proteins are highly controversial. NLRH1, you get yelled at every time you talk about NLRH1 because some people think it's pro-inflammatory, some people think it's anti-inflammatory. Everybody in the field is mean. They don't get along, and so it's quite controversial as to what it actually does. And so we're all left in the middle trying to do our best to figure out the pathways. So we're hoping that computational modeling can help give us a better definition of what these NLRs and TLRs can do. All right? And again, my feeling is that there's a lot of cell type specificity here. There's a lot of temporal dynamics that we don't understand. And so we're hoping that computational modeling can help us shed some light on some of the more specific aspects that we're just missing in the culture of macrophages. It makes a big difference whether we're working with alternatively activated macrophages or classically activated macrophages. Epithelial cells come in multiple flavors, right? And so we're hoping that we can investigate some of these specifics using competition. All right, so with that, I'll open the floor up for any questions. I bombarded you with a lot of information, right? So, does anybody have anything to ask? Yeah.
I was wondering if in your epithelial cell studies, did you get any chance to look at Cody's function and their impact on the epithelial integrity and immune function?
So no, we haven't. So we use epithelial cells more as a tool, and so the short answer is no, we haven't. It's a good avenue to look at, but it's just something we haven't looked at. Anybody else?
Cell culture, is there cell lines?
No, these are primary cells taken from humans. So the question is, in our epithelial cell culture, are they cell lines or primary cells? And they're primary cells. We take them from humans or from animals, from mice. Most of the time we're using mice. Human cells are hard to come by. But we've cultured them from both. They are a very nice system because they're primary cells. Now, if you just take epithelial cells out and you grow them up on a dish without doing anything else, they'll expand and they'll grow. They'll have a very fibroblast type pathology. To get them to polarize like that, you have to use transmal plates. You have to grow them on a surface with an air interface so that they polarize. So they form and meet. So it's a very challenging process, but it's one that is definitely worthwhile to do because you get the epithelial cells in their natural state rather than as a fibroblast that just spread out on the dish. Yep.
Have you ever looked at like over time, like from like neonates compared to adults, and if they have the same NLR expression and functionality?
So yes, so the question is, have we looked at expression of NLRs over time, or in TLRs too, right? So TLRs, NLRs over time in neonates and all the way up through development. And the answer is kind of, okay? We don't know a lot about these, but I'll tell you that the NLRs, most of the NLRs that are characterized maintain a set level of expression. Some people will show studies where they activate or stimulate and they get increased NLR expression. We haven't really seen that except under very specific conditions. What we think are that these are very stably expressed pattern recognition receptors. It is important that you maintain this balance. You don't want to have to rely on transcription and translation for these really important sensors. And so in a lot of cases, so the answer to your question is, yes, people have looked at it and know there's not a big difference over time in the cell types that have been looked at. And most of the cell types that have been looked at are hematopoietic reticules. Most of the people in these fields only study these receptors in the context of macrophages, to be honest with you. There's some dendritic cell work and neutrophil work in small groups, but most of the people use macrophages as their primary cell line. That being said, there are some NLRs that are critical for development. If you knock out NLRP2, you end up with embryonic development. If you knock out NF-kappa B associated genes, you end up also there with tough to work with NF-kappa B, knock out right? So they're certainly in some cases, the answer is yes. I'm sure the question probably changes. To answer your direct question, I don't know.
You look at inflammasome activation RSV?
Yes, we have. RSV is the NLRP3 inflammasome synthesis RSV. We have a manuscript in either IMBO reports of PLOS One, something along those lines. We took them, it was a battle to get published. But RSV activates the NLRP3 inflammasome. So if you don't have NLRP3, you end up getting increased levels of RSV. RSV tends to grow much faster, it tends to reproduce much faster, replicate much faster in the absence of the NLRP3 inflammasome.
And what strains of RSV?
Oh, that's an excellent question. We use 3 different strains. We collaborate with Santinal Bose for that. He's an RSV specialist. Off the top of my head, I can't remember. You can look up the papers. Yep.
So what kind of structural information are available for these receptors? And if you probe this question from computational point of view, we have to have some validation, right?
We do, yeah. So for most of the toll-like receptor, I will argue all 9 of the best characterized toll-like receptors, the ring helicase receptors, and a third of the NLRs have structural data, publicly accessible. The problem are the other ones that aren't published or aren't publicly available. The other problem is that these things are really hard to purify. Right? They have oligomerization domains. They oligomerize. They muck up these types of questions. So what you end up having are partial structures, typically a leucine rich repeat tail or the core domain, rather than getting the full length. Yep.
Going back to the RSV question, would you try to make a vaccine that specifically targeted that receptor?
Yeah, so the question is, will we try to target so in looking at RSV, will we try to design a vaccine to target the NLRP3 in this case? And the answer is no, because when we inhibit NLRP3, we actually end up getting a much worse condition. What you could design, though, are ways to enhance NLRP3 recognition. And to do that, we really need more biological information about what it's sensing. It's a big mystery what exactly it's sensing. At this point, we just hedge our bets and say some change in the intracellular environment. And that's driven by RSV infection, as well as influenza. There's a lot of parallels between RSV and influenza. Of course, they're very similar types of viruses. Yep.
So if you were to found some of the literature surrounding lower oxidative stress level
Yep.
and I'm just wondering, are there any differences between these
Yep. So the question, yes. All right. So the question is, I guess the statement is that there is quite a bit of literature looking at reactive oxygen species activation of NLR glycosomes. The follow-up to that is, is there any subtype specificity associated with neutrophils versus macrophages, for example? A lot of our work has shown that reactive oxygen species, so there's two answers to your question. So in the context of the inflammasome, NLRP3 inflammasome, reactive oxygen species production triggers inflammasome activation in both neutrophils as well as macrophages. So it's odd because neutrophils produce reactive oxygen species, but they can also be self-activated by it as well. And so there's a lot of work that we've looked at that reactive oxygen species seem to specifically activate the NLRP3 inflammasome in the context of a lot of different diseases. Flu, for example, we've shown that when we inhibit reactive oxygen species production, we don't get inflammasome activation as well. That's effective. Okay? NLRP3 inflammasome activation. And that's associated with its role in activating inflammasome. We use drugs to inhibit reactive oxygen species production in the context of flu and also several different pathogens associated with patterns like poly-IC, so virus-related agonists. Now, the reverse of that is that some of these NLRs, like NLRX1, has been shown to actually produce reactive oxygen species. This is a positive regulator of reactive oxygen species production. And so you have this system set up where activation of NLRX1 by, say, influenza or by a bacteria can produce reactive oxygen species, which then causes the activation of NLRP3 inflammasome in the production of biomarker. Does that answer your question? Sure. We have a paper in immunity. We used a dozen different ones, in vivo as well as in vitro. BPI is one, and then there's there are drugs like 2213, diazyl, whatever, another. You get them at Sigma, right? So, in general, we've been able to use basically drug targeting. But there's quite a bit of literature. There's a paper in Nature Immunology looking at loss reduction. Right?
Join me in thanking Coy for a very interesting presentation.